Okay, so again, got the handout. That means you don't have to draw the pictures, but there's still going to be stuff written on each slide that you do need to write down. You'll notice this looks just like our first slide from mitosis, except now we're tracking a couple of genes. We're tracking, and this is a fruit fly cell. We're going to call this a fruit fly because it's got the right number of chromosomes for a fruit fly, and nobody wants to deal with 46 of them. So here's 8, right? That's the fruit fly's number. And we've got 2 genes. We've got wing shape, which it turns out has a big W for normal wings and a little w for curly wings. No, a fruit fly can't fly if it has curly wings. Mutation. What do you call a fruit fly with curly wings? Fruit walk. And then we have eye color, which can be red or brown. Turns out red is dominant to brown. So that's one difference. Another difference is just your perception now, because you know about chromosomes. You should be able to see homologous pairs now, right? They were there before, you just didn't notice. Now you notice, well, there's 2 that have kind of different shades of red, and they're about the same size and shape. 2 blues, 2 greens, yellow and orange. You can see how they match up, right? Any chromosome you find in a nucleus of a eukaryote is going to have a homologous chromosome that matches it. So this is early, we're going to call this early prophase 1. You know prophase, right? Looking ahead, just know that we're going to go through a meiosis 1 and a meiosis 2. And so this is the very beginning part of meiosis 1. What color are this fruit fly's eyes? Red. They're red, because red is dominant, and this fruit fly has big R, little r, right? Big R here, little r here. Why? Now, some of you are thinking, why are there 2 little arrows? That sure looks like there's 2 big R's and 2 little R's. Anybody want to tell us why? Why we would have double the number of R's you're expecting?
Because of like splits and still needs the same genetic information.
Okay. What are you going to say?
Is each parent has there's 2 alleles?
Okay. Well, yeah, because here's one allele and here's the other allele, but there's 2 arrows for each one. That looks like 4 alleles. Why would that be? What are you going to say?
One is in the co-side, it has to split.
But you already know the answer to this. You're just not thinking of it. We said it at the beginning of mitosis. That's what he said. So what have they done before they ever even start this process? It's all copied, right? They did S phase. Meiosis is like mitosis in that way. The cell has done S phase. DNA has all been copied. So yeah, there's 2 copies of the big R allele and 2 copies of the little r allele. Okay, good. So I will tell you right now, this meiosis is easy, like easy, because you know mitosis already. It's easy, except for the next 2 slides. The next 2 slides, if you just bear down and soak them up and figure out exactly what's going on in the next 2 slides, the other 10 slides will be nothing. Okay? I don't know if it's 10. It's a lot. Okay? So, what happens next? It's still prophase 1. We call it late prophase 1. Because early prophase 1 looks just like mitosis. It's in late, at the very end of prophase 1, things start to get different. What happens that doesn't happen in mitosis and meiosis is they find each other. The homologous chromosomes pair up. They find each other. They do this thing called synapsis. That's them pairing up. And in synapsis, Because of that thing we just mentioned where these chromosomes are doubled structures, it's almost like there's 4 versions of every chromosome. We call them tetrads. Tetra is a root that means 4. But the really important thing here is a process called crossing over. What happens in crossing over, remember that this is basically 2 sets of chromosomes we're looking at, even without the replication. We're looking at this fruit fly's mom's chromosomes and its dad's chromosomes. And that crossing over means that they're actually trading pieces of each other. So I'm not very good at drawing it. I don't have all the shades I should have. But it's like they're turning into little Frankenstein chromosomes. They're actually switching and becoming kind of patchwork. What they're doing, understand, is because this is, one of these came from this fruit fly's mom, the other one came from his dad, is they're taking the genetic stuff that their parents gave them and mixing it all up. This is no longer just a mom and a dad chromosome. This is like mom-dad hybrid chromosomes. Introducing a whole bunch of variation. Yes, they're trading pieces, becoming all mixed up. There we are. Trading pieces, crossing over. Okay, that's thing 1. Late prophase 1, crossing over, takes the genetic deck that your parents gave you, shuffles it all together. So that when you deal it out into the 4 cells you're going to make, the 4 sperm or eggs you're going to make, it's not going to be alike in any of those 4 cells, and it's not going to be just like you. Right? It's going to reshuffle all that. This is part of why sex creates diversity. Questions so far? Okay. Now, what's coming next? What's going to happen next? Yeah, metaphase 1, we're going to call it. And what's it going to look like? Can you see it? What's different? How's it different from mitosis? So instead of lining up single file and getting pulled apart at the centromere, remember, they're lining up in pairs. And the thing about that is that, whichever one of these you want to call the mom or the dad chromosome, it's going to be random. There's basically a 50-50 chance which one winds up on which side of the metaphase line. That's the second thing that's going to create some diversity. So in metaphase 1, it is random which chromosome winds up on the right or the left, on which side of the metaphase line. For a fruit fly, this introduces some diversity, since there are 4 pairs of chromosomes. It's basically 2 to the 4th different ways that those chromosomes can line up, right? What's 2 to the 4th? Hang on. 16. Good. One of those possibilities, let's say heads is the dad chromosome on the right, and tails is the mom chromosome on the right. You 4, if you could just please flip that coin I just gave you one time and see what you got. So let's go top to bottom. Let's say Will's top. What'd you get? So tails, we said mom was on the right?
Heads.
So one of the configurations that's possible is this. There's 15 others, right? It's going to happen one of those 16 ways. And it doesn't have to happen the same way every time. How many possibilities would there be in a human cell? 2 to the
46. No.
2 to the 23rd. Is that a lot? Oh my gosh. Yeah, it's a lot of zeros. Like a lot of zeros. Almost, beyond the scope of imagination, how many different ways a human chromosome stack can line up. So there's your second source, right? Crossing over in the first place. These are patchwork chromosomes because we traded a bunch of mom and dad parts. Then we line the mom and dad chromosomes up randomly on the line, and boom. So from here on, nothing new. From here on, you've got this. You've got to understand our late prophase 1, where crossing over happens. You got to understand metaphase 1, where we're going to call this independent assortment, because it's sort of what explains why Mendel's pea plant genes didn't influence each other. It's because his genes he was dealing with were on different chromosomes. Now, if they're on the same chromosome, it gets different. They get linked and they do affect each other. Okay, but when they assort independently because they're on different chromosomes, we get variation like this. Okay, from here, and that's nothing special, right? They're going to separate in anaphase 1. We just did it. We just tested over mitosis. You know what's happening here. It's just slightly different because they're paired instead of single file. All the same things you're used to from mitosis are going to happen. We're going to get the butt-looking thing, the new nuclear membranes in telophase 1. Yeah, I'm not going to write all those things down because you know. But what we have now, once we get through our first cytokinesis, what we're going to have is 2 cells with, though they're replicated chromosomes, half, right? Half the normal number of chromosomes for a fruit fly. In this case, 4 instead of 8. Still replicate, still X-shaped. So we have another step to go through. And in fact, that other step, that meiosis 2, is going to look just like mitosis, just with half the number of chromosomes. So into prophase 2. Nuclei disappear, spindle begins to form. Metaphase 2, they line up. Anaphase 2, they split. So it's really those first 2, the scrambling part, right? Crossing over, independent assortment. That is everything, right? The rest is just mechanisms you already knew. What those 2 first stages do is make it so that the 4 cells we're about to get, the 4 little weird half cells that we're about to get that have half the normal number of chromosomes, haploid, 4 little haploid cells, are going to be different from each other genetically, quite different in many cases. And they're going to be different from each other because of crossing over that happened in late prophase 1 an independent assortment that happened in metaphase 1. When you are talking about phases of meiosis, you have to use the 1 or the 2, right? Or we don't know which phase you're talking about. Now, the last thing you need to know here is what these cells become. And you already know that, except that there turns out to be a difference between males and females here, at least in humans. What you get if you are a male is 4 individual sperm cells, yes. But if it's female, what we wind up with is, in fact, just 1 egg cell, 1 ovum, and 3 not eggs, 3 cells that will actually help the egg cell develop. They become part of the structure that's gonna help the egg develop and eventually become released, into the fallopian tubes and whatnot. So that's a difference between males and females. Here's a quick kind of animation showing you very simple meiosis and giving you a decent idea of the fact that these cells are becoming different from each other, from that crossing over, from that independent assortment. So you're very much taking the genetic deck that was dealt to you by your parents separately, right? You got 2 sets, 1 from mom, 1 from dad. You're taking those 2 sets and you're scrambling them all up and then dealing them out very randomly to your kids. Questions?
Why is there like or primitive, like the yellow and blue?
That's just little. I don't know. Maybe it's supposed to be kind of like the Y chromosome, little stubby thing. Good. So let's give this some life. Let's, instead of just looking at it on slides. And this is a little seasonally inappropriate now. Usually we get to this sooner, and it's a design your own elf lab. Because elves, Christmas. Let's call it a design your own humanoid. You pick your mythical or non-mythical humanoid critter to design here, but there's some genes that it's going to have that we're going to dictate. I'm going to give you some dice. This is the parent elf or humanoid, the parent humanoid you're going to be creating today. Rolling some dice. Sure. Yep, I do that. All right, so before we do any kind of meiosis stuff here, which will happen tomorrow, you got to have humanoid parents. So you're going to roll some dice and just randomly determine the genotypes of this humanoid. Remote folks, if you don't have dice at home, I've posted a virtual dice link for you. You're going to get 2 dice from me, and for each trait of this humanoid, for example, earlobes, and these are weird genes, weird traits, because I whenever possible, I picked traits that are simple, dominant, and recessive traits in humans, and there's not that many of those. Like, as you'll learn, most traits are more complicated than that. So I picked as many as I could, and one of them that is simple, dominant, recessive is detached versus attached earlobes. Detached means those of us that have earlobes that go down and then come back up before they go into your face. Attached is like they come down and then go straight into your head. Weirdos. Okay? So you roll your dice, and you roll 2 dice. You're going to get 2 dice. And if you roll a 2, 3, or a 4, that elf is going to be homozygous dominant. That is big E in this case with earlobes. Notice I used H's as examples at the top in the instructions. Don't be the kid that uses H for every gene. I give you a different letter for each gene. Please use the letter you're given and not just H's all the way down. Okay? So 2, 3, and 4, homozygous dominant. 5 through 9 is heterozygous. And then 10, 11, and 12 is homozygous recessive. You write down what number you roll, you write down the genotype that produces, then you write down the phenotype, then the fun part, you get to draw your humanoid. Do it on the back. Doesn't have to be beautiful. Does have to reflect the phenotypes. I do have to be able to see by looking at your humanoid what the phenotype of that humanoid is. So, lights coming at you. I recommend you just roll all the way through, write your numbers down as you go, and then you'll look it up. That way you make sure you get that right one.